So thanks for joining us today. My name is Elizabeth Crone. I'm the Head of Business Development for Research Products and Services in Europe and Rest of the World. And I'm joined by my colleague Giuseppe Ferrandino today, who is a scientist in our liver program as well. So today, Giuseppe and I will be talking to you about how breath can be used as a biomarker matrix. And specifically, we'll be talking through a case study of our own, where we've been developing breath biomarkers for liver disease to show how this can be done. To give you a bit of an introduction to Owlstone Medical first, Owlstone are a company that are ultimately trying to develop breath as a biomarker matrix. So we're doing this to enable disease diagnosis, but also monitoring of disease, including for clinical trials. And as a company, we are, of course, developing our own biomarkers. We've got very active discovery and validation activities in lung cancer, liver disease, and digestive disease. But we also offer the technology we've developed and our capabilities to academic and pharma partners who are wanting to develop breath biomarkers and test for themselves as well. And we have active studies going on with multiple partners in the microbiome space, the cardiometabolic space, and respiratory disease as well. And then finally, once biomarkers have been identified in breath and validated, one of the often required or ideal outputs would be something that can be done in the home environment. So one of the other things we've been doing recently is developing for digestive health, home monitoring sensors that can be deployed into a patient's home environment, used with an app on their phone to help them to understand their digestive health. Maybe one thing that's useful to cover before we go into any more detail is why would you maybe want to carry on listening today? Why would you want to maybe develop or use breath biomarkers yourself? People have different reasons for this, so hopefully some of these will be relevant for you. One of the ones I hear a lot is the molecules we're looking at in breath present a new range of potentially non-invasive biomarkers. It's a different range of biology that we're able to study. So this gives you the potential to identify novel biomarkers, potentially in diseases where that's been hard to do before. But it also adds to understanding of disease and maybe helping to understand different disease phenotypes as well, when layered on top of, for example, other omics. As these tests are very non-invasive, breath is a very non-invasive matrix to take. They ultimately can be much easier to deploy as tests. So a lot of people are interested in breath as a way to, say, for example, replace very invasive or expensive tests like liver biopsy or bronchiolar alveolar lavage, respiratory disease. One of the benefits of breath is that with the kind of technologies that can be used to analyze it, there is a potential for real-time readout. As you heard me say on the previous slide with the digestive health tool we're developing, there's a potential to have sensors that can be used to give a real-time readout of levels of these molecules on breath, giving the potential to, for example, give rapid diagnosis of acute conditions. And alongside that, home-based tests could, of course, also be developed where either they're using kind of sensing technologies as that breath sensor is going to be used for digestive disease, or alternatively something where patients can breathe into tubes and send them away. So this can be used, for example, for home-based monitoring of patients with variable diseases. So, for example, patients who have liver cirrhosis, for example, and need to be followed up to see if they're entering decompensation or something like that. It could be something where decentralized trials could also be enabled, giving the potential to get very rich data from studies with very frequent patient sampling, but at lower cost without having to have them go to a clinical center. And then finally, something we're considering at the moment, screening tests in the community as well. And then finally, one of the really nice things about breath is that because it's non-invasive, because it's very easy to take, it doesn't take very long, you can actually enable serial sampling over something like minutes to hours as well. So this is something that's very relevant in our liver disease program, as Giuseppe will talk about. But this allows you, for example, to use breath samples to explore, for example, how different foods or drugs are being metabolized in different people, or maybe to assess the very early activity of rapid-acting drugs. So I'll hand over to Giuseppe now to talk a little bit about what kind of biology we're looking at in breath.
Thank you, Liz. Hello, my name is Giuseppe. I am a lead translational scientist at Owlstone Medical working on the hepatology program. So, volatile organic compounds that we can measure in breath can be generated by several metabolic processes. For example, lipid peroxidation can generate aldehydes, alcohols, or alkanes. Fatty acid oxidation can generate ketones such as acetone and maybe also 2-pentanone. And then we also have VOCs generated by microbial metabolism such as short-chain fatty acids, indole or 2,3-butanediol, and several other metabolites that we will see later. However, VOCs can be also generated by exogenous compounds that get into our body through exposure, for example, like through smoking or using perfumes or pollution or exposure in the workplace. Also, with our diet, we ingest several VOCs. For example, if we drink a glass of orange juice, we are ingesting terpenes that are exhaled in the breath. And we will talk about limonene, which is a terpene that is metabolized in the liver. There are some VOCs also generated from drugs. Now, overall, what these VOCs represent? So, they are the product of metabolic processes in our body, and therefore, when there are alterations to these metabolic processes, their abundance may change. And therefore, we can use these differences to identify subjects with a specific disease when by using the alterations of these metabolic processes. So, for example, if we look at the liver, so it is a source of many VOCs. So one of these is of exogenous origin, like limonene, which is metabolized by the CYP system to compounds that then can be excreted through the urines. And then what we see is that when there is cirrhosis, limonene is reduced. There are also several other metabolites which are generated in the liver itself, but also with the crosstalk between the liver and the gut, or the liver and the adipose tissue. So one example of a volatile generated from the gut is the trimethylamine, which is converted from choline by the gut microbiome. And then TMA is further converted to trimethylamine N oxide in the liver. Now, the gut microbiome is also a great source of VOCs. And usually the microbiota feeds on the compounds that we ingest with our diet, and they produce these metabolites. For example, carbohydrates can be fermented to ethanol or 2,3-butanediol, and these metabolites have been found associated with non-alcoholic steatohepatitis. Or another example is tryptophan, which is converted to indole, and we found that this compound is elevated in subjects with cirrhosis. Amino acids can also generate VOCs that can be associated with the inflammatory bowel disease or even neurodegenerative diseases. So just to focus on some example, here we can see the endogenous ethanol production in subjects with non-alcoholic steatohepatitis. So in this experiment, the authors treated the subjects with 4-methylpyrazole, which is an alcohol dehydrogenase inhibitor. Then they gave a carbohydrate challenge and collected the blood at different time points and measured the ethanol in the blood in the subjects. As we can see here in blue, subjects with NASH have quite a remarkable spike of blood ethanol after carbohydrates intake. And then they estimated that these subjects may be producing up to 100 grams of ethanol per day, which is a considerable amount of endogenous ethanol production, more or less the amount contained in a bottle of wine. So another example is the choline TMA-TMAO pathway. In particular, trimethylamine N oxide has been found associated with cardiovascular diseases. And then at the preclinical level, TMAO has been found to increase the cardiovascular lesions in mice, suggesting that this other pathway may also be involved in exacerbation of cardiovascular diseases. Now, of course, measuring these compounds in blood is feasible. However, it's not very compliant for the subjects. And the translatability to a breath to a diagnostic test for these abnormalities in the gut is low because of the difficulties in collecting the blood at different time points. However, ethanol and trimethylamine are volatile compounds detectable in breath, and therefore we can use breath instead of blood to identify those subjects that have these excessive production of these metabolites and characterize the effect of these metabolites on the exacerbation of the diseases. I will hand over to Liz to talk about how we measure these VOCs in the breath.
Thank you, Giuseppe. So to enable the identification and development of good breath biomarkers to help us to work out which are the right VOCs to analyze, we of course need the right technology. And Owlstone has methods to collect VOCs from multiple different sources, from in vitro samples, from mice, and of course from humans, which is where most of our technology has gone into. And we analyze these molecules by high-resolution accurate mass GCMS. So overall, our technology has been developed to minimize technical variability and noise and background that's entering the sample. That gives the ability to see, for example, smaller level biological changes and gives the confidence that actually when you're looking at a biological change, it is a genuine biological change and not just technical variability. We've also focused the technology to be able to analyze a very broad range of VOCs. And that's important when you don't know what you're looking for, of course. So the technology is optimized to give the chance of being able to look at a wide range of VOCs and hopefully pull out which ones are relevant to a particular disease. And enabling that as well is an in-house library of compounds, which makes accurate identification of the chemicals that we see much easier. This is alongside something we're developing called our Breath Atlas, which I'll talk about in a few slides' time. So to talk a little bit more about breath sample collection and that technology for humans that we've developed to be able to help us to collect very reliable breath samples. When we're collecting from humans, there are lots of different factors you want to consider when you're thinking about how to take a good breath sample. One of them is, for example, how can I make sure I'm analyzing that broad range of VOCs, some of the very high concentrations, some of the very low concentrations. So what we do is we actually collect onto what's called a sorbent tube that allows us to pre-concentrate VOCs from a breath sample that's typically a few minutes long. And this allows us to be able to see both the higher concentration VOCs, but also the lower concentration VOCs. We also have to think about how to minimize background. There are VOCs in the air around us, and of course we want to prevent those from coming in and interfering with samples. So one of the things that we have as part of this breath collection station, which you can see in the center, is this Casper system, which filters out background VOCs from the air, allowing the patient to breathe in a cleaned up air supply. And so the sample we're collecting of the patient has minimized levels of the external VOCs present. Alongside that, the actual materials that you build sampling collection systems from can absorb VOCs, but also leach VOCs. So the materials we've used to actually do this breath sampling collector and develop it have been optimized to minimize those kinds of effects. And then finally, of course, you've got to actually think about how to collect your breath sample in such a way that's going to be reliably comparable with another one. So we collect a fixed volume of breath. And the receiver is plugged into a computer, which essentially allows you to monitor how a breath collection is going, program in a particular volume of breath that you want to collect, and also program in a specific fraction of the breath. We typically focus on the lower breath fraction that's been in more recent contact with the bloodstream. And so it's going to contain a much higher concentration of VOCs that are biologically relevant versus things that you're just inhaling, basically. So the receiver is computer guided. It gives information to sampling staff to tell them if they need to change anything about how the patient's breathing or anything like that. And it's designed to be able to be used across multiple clinical sites, minimizing any variability between sites as well. So another thing that Owlstone's doing to enable breath biomarker development at a faster, more rigorous pace is developing an atlas of compounds that are typically found on breath. And by on breath, I mean a compound that is clearly distinguishable from background noise. So by background noise, I mean a sample of air that's been taken that the patient is basically breathing in. So it's what we refer to as a system blank. So Owlstone are basically running a project to map and identify the on-breath compounds that we see across different studies. We're calling this the VOC Atlas. This VOC atlas is basically now a growing list of compounds, which we found on breath, typically in a healthy population, but that's expanding. And it's got quantitative levels of each of those molecules from previous human studies. What this allows us to do is basically focus in on VOCs when we're analysing the data that we think are more likely to be potential VOCs. And it also gives additional information on where those VOCs have previously been seen, if, for example, they're correlated with gender or age or other potential biases, and also whether they've been seen, for example, in similar diseases. So Atlas, we can target in our GCMS data. So we're able to give quantitative data on the compounds in our Atlas with our GCMS, alongside doing a broader untargeted analysis of the other VOCs with identification aided from a library of compounds. We're also running on our instruments called our HROM library. So to give you a bit of a clue about the diversity of biology in this Atlas and what's in there at the moment, we've actually been quite surprised, I think, by the diversity of molecules we have seen in the human healthy breath. So we've seen up to 150 VOCs so far that we've conclusively identified and know exactly what they are. And it looks like from those compounds, about 10% seem to have microbial origins. 52% look to be coming from human metabolism. So that could be, for example, lipid peroxidation, inflammatory pathways, nutrient metabolism, and things like that. And we've also got 37%, which are exposure related. So things like compounds you're taking in through your diet, from industrial processes, from smoking, and things like that. A lot of the VOCs that are in our atlas have also been associated with multiple different diseases. So you can see here's an illustration of some of the VOCs in our atlas being associated with respiratory disease, with cancer, with metabolic and GI disease, and with infectious disease as well. So I'm going to hand over to Giuseppe now to talk a little bit more in detail about how this technology is being used in the liver disease program so far.
Thank you, Liz. So what I'm going to show you are results about detection of cirrhosis from the breath using biomarkers that are altered when this condition is present. So here we can see a schematic of the disease progression. So usually the liver accumulates fibrotic tissue, and then until we reach the end stage disease, which is cirrhosis. So often these stages are asymptomatic until the subjects undergo decompensation, and therefore they need to be carried to the hospital, and there they get a diagnosis of cirrhosis. There are reports stating that up to the 75% of the subjects with cirrhosis are detected at this stage with decompensation. So therefore, detecting cirrhosis earlier will be beneficial to start treatments and preserve better liver function. So to obtain a proof of principle that we can identify subjects with cirrhosis using a breath test, we used the breath biopsy Omni and perform a targeted analysis looking at limonene with absolute quantification, and then an untargeted analysis to look also for additional compounds. Here we can see the subjects' characteristics. We had 42 controls and 46 subjects with cirrhosis, of which 14 were complicated by hepatocellular carcinoma. So we can see the median MED score was 8, and most of the subjects were Child-Pugh A or B. So here are a summary of results. So we found several compounds upregulated in the breath of subjects with cirrhosis, including limonene, and some compounds downregulated. When we combine these compounds in a classification model with cross-validation, we can see that we measured an AUC of 0.95, suggesting that we could identify efficiently subjects with cirrhosis using a breath test. So we further explored the correlation of these VOCs with the blood metrics, namely INR, albumin, and bilirubin, which are used as a proxy of liver function. And for example, we can see here that limonene correlates positively with bilirubin and negatively with albumin. So we explored these correlations further using a canonical correlation analysis, and we can see that the blood metrics, INR, albumin, and bilirubin, correlates with the breath VOCs in the first dimension. And if we perform a principal component analysis of these VOCs, we can see that we observe a separation on the first component of the cirrhosis subjects compared to the healthy controls, which are represented by the empty circles, especially if they have a higher Child-Pugh score, indicating that they separate based on the disease severity. So now, why we focused initially on limonene? So, and that's because limonene was identified already since 1994 in different studies comparing the breath of healthy subjects against subjects with cirrhosis. The proposed mechanism for increased limonene is that it is metabolized in the liver by the CYP enzymes, and so the activity of these enzymes is reduced, and therefore amount of limonene in breath increase in subjects with cirrhosis. So besides looking for the biomarkers with the best discriminatory performance, we also investigated biomarkers and see how they could be used for a breath test that is generalizable to the population. So in particular, we can see here on top the classification performance of biomarkers identified in a cohort of subjects collected in Chile, comparing 29 cirrhotics against 29 controls. We can see an excellent AUC of 0.98 in the training set, and we included these best performing biomarkers. However, when we tested this classification model on a UK cohort represented by 12 cirrhotics and 12 healthy, we found suboptimal classification performance with an AUC of 0.76. Probably this result is because some of these biomarkers are generated by local exposure and therefore may not be so useful for a diagnostic test because they can be applied only to a specific location. So to overcome this problem, we selected biomarkers that were present in the VOC atlas, suggesting that they are more widely abundant in breath, and therefore they are more generalizable to multiple populations. Indeed, if we build a classification model based on this approach, we can see now an AUC of 0.90. However, if we externally validate this model on the UK cohort, we can see now an AUC of 0.85, retaining excellent classification performance, suggesting that implementation of biomarkers that are present in the atlas and therefore more common in breath is more useful for generating a test that is generalizable and therefore useful in the clinical reality. Moving on the volatile organic compounds for liver disease. So we tested a different approach. In particular, so we used the exogenous volatile organic compound or EVOC probes to assess liver function. So we administered the compound and then we measured the compound itself or the hepatic bioproduct. This approach relies on the notion already well established of the pharmacokinetics of drugs in subjects with cirrhosis. What is reported is that compounds with high hepatic extraction show increased levels in the blood of subjects with cirrhosis compared to healthies because the perfusion of the liver is impaired and therefore these compounds remain in the blood for longer time. However, compounds with low hepatic extraction are not so much affected by the perfusion, and therefore they show a retarded decay which is due to an alteration of the enzymatic activity in the liver due to the presence of liver disease. Now, another advantage of this approach is that we can mimic the pharmacokinetic, but just measuring the concentrations in breath. without the need of collecting blood. So which increases the compliance for the subjects. And we can use compounds that are generally recognized as safe, GRAS, that those are used as flavoring agents, and we are exposed to them on a daily basis, and we don't need to run phase 1 trials to administer them to subjects. So now, if we look at the alterations of the liver in cirrhosis at the pre-cirrhotic stages, we can see that, for example, in this study, that liver perfusion is altered already at earlier stages of fibrosis. And in particular, the authors here measured the average vessel area, and we can see that starts to go down as the fibrosis progresses, and also the perfusion speed, which increases as the fibrosis progresses. Meaning that if the blood flows faster to the liver, then there is less time for the exchanges between the blood and the hepatocytes. So now to assess these notions, so we conducted the study in Chile where subjects were asked to fast overnight. Then we gave a standard dose of limonene, and then we collect the breath before and at different time points after limonene administration. So we got the breath biopsy cartridges and quantified limonene using GC-MS. So this is just a snapshot of the results. As we can see here, so this is the amount of limonene in breath, and this is the time points collected. So 0 is before limonene administration, and we can see very low levels after an overnight fasting. So we see a spike in healthy subjects, here represented as mean, and the shaded area is the 95% confidence interval. And here is the cirrhosis subjects showing an increased amount after limonene administration, suggesting that the limonene is behaving as a compound with high hepatic extraction, as I mentioned before. So if we look at the different time points post-administration, all of them show classification performance. And in particular, here is just an example of 60 minutes, which showed an AUC of 0.9. So we also found that the limonene bioavailability is correlated with MELD, and also showed some correlation with FIB4 and APRI. Additionally, limonene bioavailability also showed a relation with the presence of signs of portal hypertension. Indeed, subjects having those signs have higher levels of limonene in breath compared to cirrhosis subjects that do not have signs of portal hypertension. So interesting cases were identified in this study. So in particular, here we can see this subject was enrolled as healthy. And then we can see the blue here. So they showed a limonene breath profile that was similar to that of the cirrhosis subjects. And when we asked for a follow-up ultrasound, it was found that this subject actually had a liver disease, and they discovered it by enrolling in this study. Another subject was enrolled as cirrhotic, but the limonene breath profile was similar to that of the healthy controls. And then we asked a follow-up ultrasound, and it was found that this subject responded very well to treatment, to causative treatment, and they showed cirrhosis regression. So now, although these are only 2 cases, but this suggests that a limonene breath test could be used for detection of subjects with cirrhosis and also follow disease progression and regression after therapeutic interventions. So we also investigated some preclinical models to see the metabolism of the EVOC probes at earlier stages compared to cirrhosis. In particular, we used the model from CN Bio, where primary hepatocytes are cultured on a scaffolding, where they can be kept differentiated, and a NASH phenotype can be induced. Indeed, here we can see increase of NASH biomarkers, such as TIMP1 and interleukin-6, that are elevated in NASH hepatocytes compared to healthy hepatocytes. So once the phenotype was established, we treated the media with the EVOC probes and collected the media at different time points. So, and these are just some representative results. So we can see a substrate that is going down over time as expected because it's a volatile compound. However, if we look at the bioproduct of this substrate, we can see that it's not increasing in the control where there were no hepatocytes. However, in the presence of hepatocytes, we see an increase. And the profile is different in healthy hepatocytes where we see a spike followed by reduction, while in NASH hepatocytes, the increase is slower and steady over up to 24 hours. So this result indicate that the EVOC probe approach may be used also to identify metabolic differences between healthy subjects and subjects with non-alcoholic steatohepatitis, or also MASH as it is called now after the change of nomenclature. So moving on from the liver, we looked also at metabolites generated in the gut, as I showed you before. In particular, so we have seen already this experiment. And so we posed the question if we could translate this approach, instead of measuring ethanol in the blood, we can measure the ethanol in breath. To achieve this, we fasted subjects overnight, then performed a baseline breath collection, followed by glucose administration, and then post-administration breath collection up to 90 minutes. So here we can see 10 healthy subjects that we enrolled, and we found that after glucose administration, all the subjects showed a spike in breath, and we can clearly see compared to the baseline here. Although some early spikes around 15-20 minutes are probably ethanol generated from the oral cavity. So in conclusion, we identified the breath biomarkers for cirrhosis that correlate with disease severity and have the potential to be generalizable to different populations. The EVOC probe approach yields better classification performance for cirrhosis compared to a steady-state measurement and could be applied at pre-cirrhotic stages. Breath analysis can be used to explore the effects of microbiome-derived metabolites on disease progression. What we are going to do next is to deploy a breath test for the identification of subjects with cirrhosis before episodes of decompensation, extend the EVOC probe approach at pre-cirrhotic stages, such as metabolic dysfunction-associated steatohepatitis, MASH, according to the new nomenclature, and then characterize the effect of microbiome-derived metabolites on the disease progression and exacerbation. So now I'm going to show some results of the OLIVE study, and an internal study that we conducted at Owlstone Medical, looking at breath biomarkers in subjects with diabetes. So there were previous evidences that diabetes can change the breath profile of subjects. And in particular, so some of these metabolites can be generated by cell metabolism, by alterations in the glucose homeostasis, by the microbiome, or by the increased oxidative stress. So in our study, we enrolled 29 healthy controls, 20 subjects with pre-diabetes, and 27 subjects with type 2 diabetes. And we can see here the characteristics of these subjects. So they were asked to fast for more than 2 hours before breath collection, and then we collected just one breath sample and analyzed it with breath biopsy omni to identify features that could be discriminatory between the different groups. So we found some short-chain fatty acids, for example, acetic acid and isovaleric acid, that were increased in subjects with type 2 diabetes. 3-methyl-indole also was increased, which is a compound originating from the gut microbiome. Additional compounds that were identified were methylpropyl ketone, also known as 2-pentanol, 3-pyridinol, gamma-undecallatone, and another unknown compound, but the last three, the identity still needs to be confirmed. So those are just tentative IDs. But anyway, so these results show that there are VOCs that show an alterations in subjects with type 2 diabetes. So just to summarize, statistically significant compounds may have been generated by alterations of the gut microbiome or may have been generated by exposure. And in particular, this latter could be evidence that the subjects with type 2 diabetes could not metabolize these compounds as efficiently as healthy subjects. Now, I will hand over to Liz to talk about additional aspects of breath analysis.
Thank you, Giuseppe. So I hope that was a useful overview for you of how our technology and how this development of breath biomarkers could be useful for you. One of the things I just wanted to touch on to close off is, of course, the technology we've been using so far has been very much focused on a research setting. It's technology that's designed to help us to really thoroughly identify and validate biomarkers so we can be sure on the molecules we're taking forwards. But of course, that's not where we want to leave this. Ultimately, one of the benefits of breath, as I said at the beginning, is that it could be used, for example, to enable at-home patient monitoring. So it's important to say that once biomarkers have been identified, there's another step here, which is development of kind of fit-for-purpose assays or tests. And that's really going to depend on what the VOCs are that have been identified, so what the concentration is, what the chemistry is, but it's also going to depend on the user requirements. So for a particular test, what's the clinical need for it? How quickly do results need to be given? Where's the collection needing to be taken? Is it needing to be done in the home environment? Is it needing to be done in a hospital environment? And then finally, of course, the commercial requirements. What are the commercial limitations on a test to make it actually something that could be useful and viable in a healthcare system? So Owlstone are already working on this. It's this back end of things as well to enable the technology to be there. So when VOCs have been identified, they can be translated nice and quickly and easily to something that's useful to patients and clinicians more directly. So we already offer and perform CE-marked digestive disease tests for the NHS when referred to by specialists, things like SIBO and carbohydrate intolerance. But as I said, we're also developing this at-home system, which is actually going to be measuring hydrogen and methane on a patient's breath that can be used in their home environment. So to show you a little bit about what that looks like, this is a device we're talking about. It is an at-home monitoring system for digestive health. So it gives real-time readout of hydrogen and methane levels. The app also contains multiple questions and things for the patients to input data into, so that ultimately we can correlate the data and spot trends and correlations with for example, symptoms and hydrogen and methane levels, and help them to manage their health in a more active and a proactive way. So how can Owlstone help you with your research and with perhaps integrating breath as a matrix that you might be interested in applying in your research? The first, of course, is to enable the discovery and characterization of relevant molecules in the breath that might be useful for the particular research you're doing. So we offer several things that can help with that. Firstly, of course, is some methods to actually enable the reliable collection of VOCs from multiple sources. So from in vitro sources like tissue culture, from rodents, and also from humans. We offer the analysis of these molecules in the world's largest breath analysis laboratory. We can give quantitative analysis on a list of on-breath VOCs that we've already seen in a healthy population. We've got the comparable levels of those molecules in our atlas. We can also do untargeted analysis to identify completely novel molecules as well. A lot of the stuff that Giuseppe has been talking about in this presentation, with these EVOC assays, for example, for the liver enzymes, we can also offer those assays for use in your research as well. So if this is a particularly interesting area for you, and you've got thoughts on how you'd want to kind of deploy those EVOC assays for yourselves, again, please feel free to reach out. It'd be great to have a conversation with you. And then finally, we offer the support in the actual analysis of these data. So the Atlas and our team internally help us to enable biological interpretation of the findings and support in further development of characterization of these biomarkers. For example, if we see interesting molecules in an untargeted analysis, like we did in our liver disease work, we could hypothesize it where they're coming from, the enzymatic methods that they use that they're actually generating them, and potentially even develop EVOC assays for those kinds of things as well. And then, of course, once VOCs have been identified and characterized, we can work with you to help develop and deploy those either for large-scale clinical studies or for actual clinical use. So that could be, for example, home-based breath collection with central lab analysis. It could be point of care devices where immediate readout is needed, or it could be in a clinical environment where the patient's in hospital already and they're going to need a breath collection, again, with central analysis, lab analysis potentially. So thank you for joining today. I hope you found that useful. If you're keen to explore more how to deploy this kind of technology into your own research, there's a few different options for you. First, there's our breath biopsy guide, which is available online and summarizes our technology and quite a lot of the recent research in breath biopsy. There's also our breath biopsy conference, which we hold yearly. This year's conference is going to be on the 5th and 6th of November. And you're able to submit abstracts for that, I believe. At that conference, that's where we talk about the latest breath research that's gone on in the field. It's a great opportunity to hear kind of the diverse range of work that's going on. Or, of course, you can reach out to have a chat with us directly to discuss clinical studies of interest for yourself or to just generally explore this a little bit more. If you're in Europe and rest of the world, then I'm the right person to talk to. That's my email address there, elizabeth.crone@owlstone.co.uk. Alternatively, if you're in the Americas, my colleague Hugh Davies is the right person to talk to. His email is hugh.davies@owlstone.co.uk. So thanks again for listening today. I'm happy to take any questions.
So my name is Hugh Davis. I'm heading up the commercial team here at Owlstone for our breath biopsy research products and services. I'm going to field the questions today. Just a reminder, if you have any questions, please put them in the Q&A box on chat. Any questions we don't get a chance to answer today, we will post afterwards and you can watch those on demand. So I guess a first question probably for, is Giuseppe on the line as well?
Yeah, I'm here.
Hi, Giuseppe, yeah.
Hello.
What are your plans next for the liver test, if you can sort of briefly summarize that?
Yeah, so the advantage of breath analysis is that so it's a non-invasive method and we can develop tests that are fairly easy to administer compared to the available testing where you need well-trained and specialized people. And therefore now we are aiming to develop a breath test for cirrhosis, given that we can differentiate those subjects very well to establish in primary care if they need further follow-up for liver diseases or they need to look for other causes of the symptoms. And then, of course, like after that, we are looking also to implement and see how the approach works in earlier stages of liver disease, such as metabolic associated steatohepatitis.
Great, thanks, Giuseppe. Related to the diabetes study, could you comment on why you think short-chain fatty acids might be elevated in diabetics?
So yes, there are some hypotheses on why these go up in diabetics, and so we found this is in breath, but it was found also in blood. So one hypothesis is that there is alterations of the gut microbiome, which coupled with the leaky gut increases the passage of short-chain fatty acids to the bloodstream, and therefore we can see an elevation in breath. So usually short-chain fatty acids are widely used by the gut epithelia. However, so probably in diabetes, this process may be impaired, and therefore more of these go into the bloodstream.
Perfect, thank you. And then again, a question for you, Giuseppe. It's a three-part question, really. How can you be sure that limonene evoke is not merely being transmitted from the GI tract to the oral cavity? So perhaps answer that one first. There's a couple of other related questions here.
Yeah, so we developed a formulation, an emulsion actually, which has a high bioavailability such that limonene is quickly absorbed. So if we use, for example, limonene oil, which forms big droplets when it gets into the stomach or in a liquid, because limonene is quite hydrophobic. What we observe is that we get a signal after 5 minutes, which is probably signal generated by the oral cavity, and then limonene disappears. And then it comes up again after 1 hour. And of course, that's because the MR limonene oil is absorbed more slowly given the droplets size. So yeah, so you were mentioning is a multiple question?
The follow-up is actually, I think you addressed them in your answer there. Limonene, is that in a capsule or other formulation? And also the time points measured seem quite quick for absorption and exhalation on breath. So I think that was probably included largely in your answer there. Question for you, Liz. Have we ever done any work on animal models?
Yeah, very good question. I think looking into the Q&A there is animal diseases as well. So in terms of animals, we have recently developed a setup to allow us to sample from rodents that we're able to use in research with people. So that can be used across a wide range of different types of diseases, but it's specifically, I guess, used for generally more animal model studies of human diseases. I guess you could use it to look at animal disease, but it is currently used for rodents. And I guess there's not too much interest in looking at rodent diseases in the broader research area.
Thanks, Liz. Probably another question for you, Liz. How stable are the breath samples after they've been taken?
Yeah, so the way we take these samples, actually we're sampling them onto these tubes here that you can see. So when a breath sample is being taken, the VOCs are pumped across these tubes. The rest of the air is expelled out through the end, and the VOCs are basically captured on a sorbent material that's inside these tubes. Once those are taken, we would receive those back to our lab, and then we do a process called dry purging. Dry purging removes the water from the tubes, which vastly improves the stability of the compounds. There will always, of course, be some very reactive compounds that do react and decay almost instantaneously. But a lot of the compounds that we're looking at are stable for a longer period of time. And again, if you're doing a longer study, one of the things that we can do, for example, is analyze the individual compounds and do data analysis to try and work out what any breakdown of those compounds might have been, to be able to help us to factor in that into any of the data analysis. But broadly, what we see is the compounds are relatively stable when they're trapped onto those sorbent materials.
Thanks, Liz. One for Giuseppe here. Are there any diseases that could affect metabolism and the results? And in particular, do respiratory or other obstructive pulmonary diseases affect the results? Any thoughts on that, Giuseppe?
Yeah, so this is something that we need to address in further development. So initially in our trials, given that they were proof of principle studies, so we excluded subjects with lung problems. But now we are progressing with the larger studies in which we enroll also, so we don't put this exclusion criteria. Hopefully we will get a sample size of people with this problem that will allow us to understand if the breath test could be administered also to them.
There's a question here, I think. Do you have experience in detection of HCN? I guess that's hydrogen cyanide, is it, in breath? And by what technique?
Yeah, hydrogen cyanide. So if it is a volatile, we can definitely look at it and see if we can measure it in breath. Yeah, and then if there is an interest in a specific association with the disease, then we can see what we can do.
Perfect, thank you. One for you here, Liz. Is pre-concentration necessary for VOC analysis, or are there direct sampling methods as well?
Very good question, and it's all going to depend on which particular VOCs are relevant for us to look at. So if you look at VOCs like acetone and isoprene, they're typically very high in abundance, so you don't have any problems with the analysis on the back end of those. You could read those out quite quickly. It's also a combination, of course, of what the sensitivity of the analytical equipment is. So if you're working with very sensitive analytical equipment, you might well not need to concentrate up a breast sample, whereas if you're working with lower sensitivity equipment, you might need to concentrate it up further. What we do know about VOCs is there is a really wide range of concentrations of these molecules. So in most of our studies, they're more focused on trying to understand what are the relevant compounds that we're seeing that might be changing between different disease groups or between different time points in a study. And for those kinds of studies, we really want to be able to analyze as broad a range of molecules as possible to try and help us to work out which ones are the right ones to look at. So for that, what we want to be able to do is not discount anything that's low abundance. So we would typically want to take a fairly concentrated, longer duration of breast sample to help us to concentrate up those really small abundance molecules, as well as helping us to see the higher abundance ones. The GCMS that we do on the back end is a very wide dynamic range, so it can cope with quite a wide variety and concentration.
Thank you, Liz. A question here about the material we use in the sampling tubes. Could you comment on that, Liz?
Sure, yeah. So it's a dual bed tube that we're using. I can't reveal the particular sorbents, unfortunately, but what I can say is it's optimized to capture, again, that very broad range of compounds. So it's got some material in it that captures the higher volatility compounds, but it's got a matrix in before it reaches that high capture one, which has a lower absorbent capacity. That means it captures the lower volatility compounds that are typically higher molecular weight. So those are basically captured on this initial sorbent bed, that then goes through into the higher capacity sorbent bed that captures the kind of higher volatility compounds. So it enables us to capture a fairly broad range of volatiles. We see generally something between carbon 3 and carbon 15 up to carbon 20.
Okay, question here. Many organs can contribute to the composition of breath. How can you be sure that particular breath biomarkers come from specific organs, those that one is interested in? I'll let you have a crack at that one first, Liz, and maybe Giuseppe has some thoughts as well.
Sure, yeah, I'm sure we can tackle that one together, Giuseppe. It's a really interesting question. It's actually one we get all the time. The short answer is it's really very challenging. So when you're looking at a breath sample, we actually typically collect the lower fraction of breath because that's the fraction that's been in most recent contact with the bloodstream. So we see a higher abundance of volatiles coming in there that generally have lower variabilities on them. So what we're looking at in that, of course, is anything that's come from the wider bloodstream. So anything that's in contact with the bloodstream can potentially send VOCs into the bloodstream that can then be seen in breath. So in short, it's actually very hard to distinguish that. I think Giuseppe's done some fairly clever work when he's looking at the EVOC side of things to help us work out if we're seeing things that are coming from kind of the oral cavity rather than the bloodstream. Maybe you could talk through that, Giuseppe?
Yeah, sure. Yeah, for example, so basically when there are disease conditions, so there are metabolic alterations, therefore pathways are upregulated or downregulated. And then we can see those alterations in the breath. The liver is a nice example because we have several metabolic pathways that happens mainly in the liver. And therefore, yeah, there is a small fraction also happening in the kidney mostly, but so basically that's a nice example of where we can isolate specific metabolic processes. But of course, like for example, in cancer and in other diseases like fibrosis of our tissue, I'm not an expert of those other diseases, but there are metabolic alterations in those conditions as well that can cause changes in the VOCs in breath. And yeah, there are lots of papers showing these alterations in those populations.
Thank you. Giuseppe, probably one for you here, if you know the answer to this. So what is the minimum concentration of biomarkers in the breath that could be detected by a device to determine liver disease and type 2 diabetes? I think probably in the context of limonene, you maybe have some ideas there.
Yeah, so we measured in terms of limit. So we have like a limit of, we established the limit of detection and limit of quantification. And we are quite, yeah, basically we are in the nanogram per microliter range, sorry, nanogram per liter of breath range. so which is sensitive enough for us to differentiate the subjects. So yeah, so in terms of how much we need to measure, so basically now we are conducting larger studies to establish the thresholds which show the normal levels in healthy subjects, and then the threshold for subjects with with cirrhosis, for example. So we have an idea for now. So basically, for example, after an overnight fasting, so healthy subjects show levels of limonene in breath that are quite low, fairly close to the limit of quantification or the limit of detection, while cirrhosis subjects show higher levels.
And I guess to add in there as well, it's important not to just look at the breath samples for that quantification level, but also the blank samples. If someone's been breathing or there's compounds that are similar in the breath as what are in the environment, you also want to be looking at the environment sample as well, and making sure you see a signal above that background level in a blank sample.
Oh yeah, of course we collect always blank samples to show that, to make sure that what we measure is coming actually from the breath. And indeed, as I showed before the example, so not always the best performing biomarkers are the one, so the best one to pick up, but so it's better to pick up others that are more generalizable and have higher chances to be used in a breath test.
Great, thank you both for those comprehensive answers. I think we're drawing to a close here now. So I'd just like to thank all attendees for joining the webinar today. I'd also like to thank Liz and Giuseppe again for their time and their excellent presentations. If you'd like to watch the webinar or look at some of the, look at some of the questions we didn't have time to answer today, you'll receive the links to those in the next couple of days. And as Liz mentioned, we also have a breath biopsy conference that's going to be on the 5th and 6th of November this year. It'll take place online. Registration for that is open if you'd like to submit an abstract to present your latest research, in particular in the breath arena. And with that, I'd like to call the meeting to a close, and thanks everyone for joining today. Thank you.